Well, thank you so much. Yeah, it's really great to be here and to get to share a little bit about where we'd like to go. I've been running a group at MIT for about 20 years now. It's scary to think, with the goal of building the tools that allow us to make maps of the brain, control them, and to read out their dynamics. And my dream, which now is starting to, I think, maybe come into view, is can we do for biology and in particular brain science what, let's say, quantum mechanics did for physics? Can we have a list of the building blocks and how they work together that's sophisticated enough and yet precise enough that we can understand how systems work? And I take great inspiration from the path that physics had, where it not only revolutionized our understanding of the universe, but led to very practical, everyday outcomes, right? Such as the microchip. Maybe that's the most publicly facing triumph of quantum mechanics. And of course, after that matter, you have people dropping out of college and starting Apple and Microsoft and Facebook, something that was probably not going to happen before quantum mechanics. You know, all the money on earth would not build the internet before you have a microchip. So I've been starting to wonder, you know, what is the microchip moment, so to speak, for the brain? You know, right now a brain clinical trial might cost you billions of dollars and take a decade, and the failure rate, even after all that, will be like 95% failure. We want to understand our thoughts and feelings. We want to understand what it means to be human. And so to understand the brain, what we've been doing over the last couple of decades, and now we're trying to stitch it together, is to build tools for controlling the high-speed dynamics of the brain, reading out the high-speed dynamics of the brain, and making maps of wires and molecules. Now, these data sets are enormous and complicated, and so increasingly, we would like to integrate these tools so that we can build, ideally, software simulations of the brain, perhaps starting with small brains, but going as fast as possible to larger and larger ones. It might be all too obvious, but I would just mention that there are many reasons why you might want such a simulation. Of course, one of our key motivations is to pinpoint brain therapeutics. As I said earlier, brain therapeutics are fraught with risk, and even the therapies that make all their way through clinical trials tend to be partial in their effect and have lots of side effects. My greatest personal motivation at this point is probably philosophy. I've just been always, ever since I was a kid, intrigued by like, why do we do what we do, and say what we say, and think what we think, and feel what we feel. And of course, I think AI is on the tip of everybody's tongue and in everybody's mind. So the different tool sets we've been building for controlling and imaging dynamics and mapping, I'll briefly summarize, and then I'll describe how we're trying to integrate them, starting with small brains, but going in scale as fast as we can. So to control the brain, we needed a way that we could control individual brain cells' electrical activity. So we have electrical activity as one of the major ways that our brain cells compute. How do we stimulate individual ones so we can have causal information about what a brain cell contributes to, a behavior or a process or an emotion or a pathology? And so this started one called Weisroth, and I wrote with students at Stanford. We wanted to find a way to control brain cells with extraordinary precision, single cell, single electrical pulse, that kind of thing. And so we just went through all the laws of physics and decided that light might be the best way of doing it if you could get away with it, because light can be aimed very precisely, and of course, light is as fast as anything ever gets. The brain doesn't have nociceptors, so you could bring optical fibers into the brain the same way that electrodes are brought into the brains of deep brain stimulation patients all the time, even without general anesthesia. Then the question becomes, how can these delicate tiny brain cells convert light to electricity, right? We need to shine light on a cell and get electrical activity. And so I became fascinated by microbial rhodopsins. These are proteins that look a little bit like the thing in the lower left. They sit in the membranes of single-celled microbes where they convert sunlight into electrical signals that then power ATP production and other kinds of bioelectrical and biochemical signals. So the first to be found were light-driven proton pumps from single-celled microbes that live in salty water, like in the upper left. I think they were actually first discovered here in San Francisco Bay. People later found pumps of other kinds of ions and channels that pass positive and negative charge and so forth. And all over the tree of life, we can find these rhodopsins, which at a glance look a little bit like the ones in our eye, but their details are quite different. And so to make a long story short, what we found is that members of all these classes can be found that are fast enough, strong enough, and safe enough that we can express the genes that encode for these proteins in brain cells, shine light on the brain cells, and be able to control their electrical activity. So by triggering a neuron, we can see what is sufficient to initiate, and by silencing a neuron, we can find out what it's necessary for. More recently, we've been exploring how to do holographic stimulation, holograms for the purposes of this talk, you can think of as three-dimensional light sculptures for the brain. Can we stimulate brain cells one by one? What some of my colleagues like to call playing the brain like a piano. And can we dial in computations so that we can really understand how entire ensembles of neurons work together? So we're working on adapting the holographic idea, which is pioneered by people like Valentino Miliani and others, so that we can try to ideally control entire brains, starting with small brains, but growing as fast as we can, with individual cell resolution. That's control. What about readout of brain activity? So we've been building what might be some of the world's fastest microscopes, taking advantage of tech development trends like the fact that cameras are getting really fast thanks to drones and self-driving cars that have much more demanding needs than, let's say, cell phone cameras and so forth. And so these are very fast microscopes. And what you see here, we're scanning a very small brain, a larval zebrafish brain, about 100,000 brain cells. And what you see here look a lot like electrical traces, but they're not. These are voltage traces. And what is happening is these brain cells are expressing genes that make proteins, and the proteins act a little bit like the opposite of those light-activated proteins I told you before. There, we control neural activity with light. Here, the neurons will fluoresce or broadcast light when we shine light on them in proportion to their activity. So these voltage traces look electrical, but they're purely optical, picked up on these fast cameras. And so this was, to my knowledge, the first example where people were imaging voltage of individual cells distributed throughout an entire brain. So if we can control neurons and image them, we also need, of course, to get the wiring of the brain. And Andrew already had introduced this concept of expansion microscopy. The basic idea is that we take a brain, preserved, not living, so this happens at the end of the experiment, and we infuse it chemically with a polymer that is a lot like the stuff in baby diapers, a swallowable polymer. And if you do it just right, then when you add water, you can swell the brain. And in our first paper, about a decade ago, we showed that we could take a piece of brain tissue and blow it up by 100 times in volume. B is a piece of the mouse brain. C, the same piece. About a day and a half later, we've blown it up 100 times in volume. And now we've been able to expand things much, much larger by changing the polymer concentration and iterating the process. Andrew teed up the point that we need to map molecules. So one thing we have been doing is expanding and then labeling molecules through stains that we can stain, image, and wash, and we can do that over many times. So these 4 images in the upper left are of 5 proteins each, stained with antibodies against these proteins, and the right-hand side shows various overlays of synapses. And you can start to see the intricate and beautiful 3-dimensional properties of these synapses. These use antibodies to label proteins, but what if you don't have an antibody for a protein? Or what if you want to go at a scale where maybe you can't economically justify that many antibodies? And so although lots of people are doing brilliant work on building better affinity tags for proteins, we've also been exploring this collaborative project led by Silvio Rizzoli, where they're taking proteins and anchoring them to the expansion polymer. You had these little anchors, and then they get attached to the polymer from many sites, as you can see in the top row of this cartoon. Then you shatter the protein into pieces and pull the pieces apart. And so that what you then see is something that looks awfully like the shape of the protein. So in gray here are sort of the ground truths, if you will, the crystal structure of a protein. And then the false color images are single, unaveraged expansions of proteins. So it's very tantalizing to think that maybe you could expand a brain, match each of the blobs that you see to, for example, its AlphaFold prediction or some other data set that allows you to infer what the molecules are, and maybe then you can read up molecular content. So the hope is that these molecular contents superimposed upon kinetomics, this is of course an image from the paper led by Andrew at E11, which we collaborated on. If you can map the molecules onto the wiring, then those 2 data sets might be very feasible for generating the kinds of maps that will allow you to infer what a given synapse or piece of a neuron is doing. And so the last slide I just want to show you is how we hope to integrate the data. In blue, from the live imaging and perturbation, we hope to build network models using deep learning of how the brain works. In yellow, with the molecular atlases, we hope to do bottom-up models where we can predict the physiology. And in the middle are these sort of middle-out models where we want to infer how to link these emergent descriptions down to their fundamental molecular recipes. And this is being done with a large and growing cast of characters that's very, very excited to work together. We're starting with very small brains like fish and worms, but our hope is to go as fast as possible to larger and larger brains. And with that, I would love to take questions if people have them.
A lot to digest in 7 minutes. Questions, comments?
Hey, James here. I'm wondering, what do you think people least understand or most get wrong when they think about the potential of the work that you're trying to do? It's a huge concept.
What people get wrong about the work we do?
Yes, or misunderstand that you wish they got. They could grok.
I think people think we know a lot more about the brain than we do. I mean, people find shocking surprises about the brain all the time. People found out some years ago that brain cells make cannabinoids, molecules that act like the active ingredient in marijuana. People found that brain cells make, in mammalian neurons, right, HIV-like particles that can then transfer genetic material from one brain cell to the other during processes like learning. I mean, these are the kinds of things that we're still discovering in this day and age. And so when we think about the repertoire of molecular mechanisms that make the brain run, the list is just staggeringly long and maybe full of unknowns. And that's one reason why I think molecular mapping and molecular interrogation are very, very key.
Yeah, we know way too little about the brain, that's for sure.
All right, thank you so much, Ed. Wonderful.